In the algae part of the syllabus, we have already studied the general characteristics, the methods of reproduction, and the classification of algae. And related to that, we have studied some of the life history, life cycles of nostoc, chlamydomonas, idogonium, and wacheria. So in the brown algae, next, that is Phaeophyceae, division Phaeophyceae, we are studying another example, Sargassum. So Sargassum is a brown algae which is found growing in the seawater and also in certain freshwater forms. And this plant or the algae, brown algae, usually it grows in the warm seas, means a sea which are having some tropical climate. And in some of the forms, means these, they grow to very large height. They appear like an angiosperm plant. So the Sargassum algae, it is, though it is brown, it contains chlorophyll, and then it as it contains chlorophyll, so definitely it is capable of conducting photosynthesis. So it contains brown pigment fucoxanthin, hence it is referred as brown algae under Phaeophyceae, that is Phaeophyta division. Now Sargassum is usually found in the warm seas attached to rocks or any other hard substratum in the water. And in this, when they are attached growing to the hard substratum like rocks, they may get separated and remain floating over the water surface. And we can observe the parts of the plant or these parts of the brown algae on the water surface. But naturally they are attached to the hard substratum, and thus they grow attached to this substratum, and they are always, they may be found in the water surface. Then this Sargassum is so called because in the Atlantic Ocean there is a sea which is called as Sargasso Sea. Now this Sargasso Sea is abundantly having Sargassum organism, Sargassum brown algae. So that is why, because of that Sargassum, the sea is called as the Sargasso Sea, and this is how the Sargassum is named. Then coming to the organization of the thallus. So the thallus is a sporophyte. So that means it produces spores, and also it produces the gametes in some parts of it. And this Sargassum, it grows to a very tall height, to about 30 centimeters in length when it is found attached. So it is attached by the help of some root-like structures, and it has a grip or like a hold fast. It attaches to the substratum. So this main axis which gets attached, it is called as the the main axis it is called as. So this is the main axis which gives rise to various type of branches like this. So these branches, they produces the lateral branches again. They appear like leaves. So these are called as primary laterals. They are called as the primary laterals or laterals of unlimited growth, long shoot of unlimited growth. They are means they grow indefinitely. It is a branch like structure, and these unlimited branches, so this they further gives rise to, so that means the primary branches, they further gives rise to the secondary laterals. So there are secondary laterals of limited growth. So these are the secondary laterals which grow on the primary laterals. These are of unlimited growth and the secondary laterals are of limited growth. So then the axillary part of this lateral growth or the secondary laterals. So these secondary laterals, they are having some swollen structures. So these swollen structures are what are called as the air bladders. So there are some air bladders which are present there. So that is the air bladder which is found. Now in addition to this, we come across some structures which are branched from the region. There we find such structures. So these structures are called as receptacles. So these receptacles are branched structures which are found, which are cylindrical and somewhat flattened, which originate in the axillary part of the these secondary laterals, and that is called as receptacles. So the receptacles contain structures called as conceptacles, which are the reproductive parts. So the conceptacles are present inside the receptacles regions, which are highly branched structures. So that is about the morphology of the thallus. Actually the plant body or the body of the sargassum is called as thallus. As we have already mentioned, whatever algae it is, whether it is green algae, brown algae, red algae, or golden brown algae, or yellow green algae, whatever it is, so all are thalloid body. That means the body of the algae is called as thallus, because there is no well defined root, stem and leaf. So here also there is no root, stem and leaf. This is not a stem, it is a stem like structure, and these are some leaf like structures. So hence, though the plant or the sargassum organism or the brown algae, it grows to about 30 centimeters in length. So 30 centimeters means almost nearly about a half meter. So hence the plant body or the body of sargassum appears like a angiosperm plant. So when we see this body of sargassum, we feel that, or we think that it is the body of or it is a plant body of an angiospermic plant. So that is how the morphology resembles the body of the sargassum resembles the morphology of the angiospermic plant, because there are some leaf-like structures and all that. But however, there are reproductive organs in the these receptacles. Then if you come to the structure of the thallus, now especially the thallus which is presented, what it contains. Actually there are 3 layers in the thallus. So these 3 layers which are present. So the thallus has 3 layers inside. So when we study the internal structure, which means it is the anatomy of the the thallus body. So here there is no question of stem, root and leaf. But when we see this, we think that this is a stem. But it is the main axis of the Sargassum body or the plant body. So as it resembles a plant, so these three layers, what are they? They are called as meristoderm. This is one layer, and the second one is cortex, and the third one is the medulla. So these are the three layers which are found in the structure of a thallus. So the meristoderm is the outermost layer. So in the thallus we find this meristoderm, which is consisting of the palisade and the hypodermal layer. So it consists of column shaped cells. So those column shaped cells, they are having abundant chloroplasts, the plastids, which contain the pigments like brown pigment and chlorophyll. So as the brown pigment is large in quantity, so it is brown in color, brown algae. And in addition to that, there are some granules which are called as the fucosan granules, and the meristoderm is covered by a mucilaginous layer called as the cuticle. That is about the meristem. So meristoderm, which is the outermost layer of the thallus. Then the cortex is inside the hypodermal layer. As the meristoderm layer consists of the palisade and hypodermis, so the within the hypodermal layer is the cortex, and this cortex consists of somewhat polygonal cells which are thick walled. So the polygonal cells are thick walled, and these because of the thick wall, they help in the mechanical function, and in addition to that, they also help in the storage function. So that is about the cortex region of the thallus. Then if they come to the medulla, which is the central part, so the thallus is having outer meristoderm, and inner to the meristoderm is the cortex. The central part of the thallus is the medulla. So the medulla has loosely arranged cells. As we have studied in the dicot stem and monocot stem in angiosperms, when we study about the anatomy, the medulla is also called as the pith. So the pith is the consisting of loosely arranged cells. There we call it as parenchyma. Here also, similar to that, the medulla, which is the central part of the thallus, it consists of loosely arranged cells which are similar to parenchyma, but we don't use the name parenchyma here. And these are having also thick walls only on the outer sides, and inner there is the thin wall. So the outer part of the wall is thick and the inner is thin, and this medulla is sometimes absent in the free floating species. So some species which are freely floating, they don't have this medulla. That is about the thallus structure. Then if you come to the reproduction of the Sargassum, how the Sargassum undergoes reproduction. So Sargassum is a brown algae. So it reproduces by vegetative method and as well as sexual method. So vegetative method, which we have already studied, it is usually by fragmentation. So vegetative method means it is usually by fragmentation, and also of course by spore formation is also there, like sporulation. But here we don't come across the production of spores, but instead of that, fragmentation is the common method of vegetation. When any part of the plant is separated accidentally, so this usually what happens in fragmentation, as the plant grows, so when the plant grows, the older parts, they die, and only the younger parts remain. So when the older parts die, so the younger parts will not also die along with the older part. Say they do not undergo degradation, they do not get damaged, they also continue to grow and form a full-fledged plant or a complete well-developed plant. That is by simple fragmentation which is taking place in Sargassum. Now the sexual method of reproduction is very important here. The sexual method of reproduction. So sexual method is oogamous type of reproduction. There are well developed reproductive organs. The male reproductive organ Antheridia, the female reproductive organ Oogonia. So these Antheridia and Oogonia, they are produced in the receptacles. So in the receptacles there are structures which are called as conceptacles. So these conceptacles, they contain the sex organs, and the male sex organ Antheridia, the female sex organ the Oogonia. So Antheridia and Oogonia, they are produced in the conceptacles. So how the Antheridia are produced and how these Archegonia are produced, we shall see. So this Antheridia which are produced, they are very small and they are somewhat club shaped structures with having two walls on the outer side and inner side. So they are produced in the conceptacles. Conceptacles, they are some structures which gives rise to the reproductive organs. So the club shaped Antheridia is produced in the conceptacle. So this conceptacle, so the outer, there are two layers here. So this outer is called as the exocyte and the inner is called as the endocyte. So these are the two protective layers of Antheridia. So the Antheridia are produced in the conceptacles, and these Antheridia which are club shaped, they are having the outer exocyte and the inner endocyte. And this endocyte is somewhat gelatinous in nature, and it is not made up of any cells there. And these Antheridia, they are produced in many numbers. So a single conceptacle, which is a somewhat cavity-like structure, it contains several or many Antheridia produced in them. So there are large numbers of Antheridia produced in a single conceptacle, and this single Antheridium. So this is a single Antheridium which is produced in a conceptacle. So there are several Antheridia, large numbers of Antheridia are produced in a single conceptacle. So this is the club shaped. Inside there are the antheridial mother cells actually. These antheridial mother cells, they divide and gives rise to the male gametes or sperms, or which are also called as the anthrozoids. So these sperms which are produced, they are actually haploid in nature. So that means about 64 biflagellate sperms are produced in one antheridium. So this is a simple antheridium which is having a two flagella like this. So such kind of sperms are produced in the single antheridium, and actually they divide by meiosis, and they multiply and they produce about 64 biflagellate sperms. So these sperms, they come out through an opening of the antheridium, which is called as the ostiole. The antheridium is having a small opening through which the sperms or through which the biflagellate male gametes, they come out. So the sperms are somewhat pear shaped, a shape of the pear fruit, and there are two flagella which are laterally inserted, but actually they are of unequal length. So if one is somewhat smaller and one is somewhat longer, so the flagella are of unequal length in each sperm, then they are released through the this ostiole, and when they come out from the ostiole, they move freely in the water and reach the oogonium, and we shall see the structure of the oogonium. So the oogonium, they are also produced in the conceptacles. Now these oogonia or the oogonium, which is the female reproductive organ, they are produced in the conceptacles, and these oogonia, they are having filled with dense cytoplasm, single diploid nucleus, and plenty of oil drops. So as the reserve food materials. So the diploid nucleus is present in the this oogonia. So suppose this is the oogonium, which is having a diploid nucleus. So this nucleus divides actually to form about 8 daughter nuclei. So it forms about 8 daughter nuclei, means it divides by meiosis and forms haploid nuclei like this. So this 8 daughter nuclei which are formed, so the 7 of them degenerate, only one will remain in this oogonium. Means it is the process of the development of the oogonium. So a single oogonium means only one nucleus will remain, and that forms the egg nucleus or the ovum, and that is how out of the total 8 haploid daughter nuclei which are formed by meiosis, so only one will remain, 7 will degenerate, and at maturity you find only one nucleus remains in the oogonium. And this oogonium which is formed, again there is differentiation of 3 layers in this oogonium. So this is the outer layer, and this is the middle layer, and there is the inner layer. So the oogonium differentiates into 3 layers, wall layers. So this outer is the exocyte, as in case of antheridium also here we come across, and the middle is the mesocyte, and the inner is the endocyte. So as the antheridium is protected by 2 layers, but the oogonium is protected by 3 layers. So when the oogonium is completely developed or at maturity, you will find the wall of the oogonium is somewhat thick. Here only 2 layers are there. When we compare it to the oogonium, the wall is very thick because of the presence of 3 layers: the outer exocyte, the middle mesocyte, the inner endocyte layer. That is how the oogonium is well protected with the thick wall. Now the sperms which are released, if you come to the fertilization. So in fertilization, the sperms are released from the antheridium, and when the sperms are released, So they move towards the oogonium, and large number of sperms, they become attached to the wall of the oogonium by the help of this flagella. So already I said 64 are produced in one antheridium. So there are several antheridia in one conceptacle. So large number of sperms will move towards the oogonium. So this oogonium gets surrounded by several number of sperms, and these sperms attach to the wall of the oogonium, and out of this only one sperm will penetrate the wall of the oogonium and enter the ovum. So though there are many sperms surrounding the ovum, and only one active sperm will get successful in entering into this ovum. And in general, we know that how the sperms will move towards the oogonium. So it is applicable to almost all organisms. Usually the oogonium releases a kind of chemical which diffuses in the surroundings, and also the sperms also releases a chemical in its surroundings. So this chemical, which is usually an hormone, it attracts the sperms towards it. So there is a kind of interaction of the chemicals. So there is chemical interaction of the chemicals which are released by the oogonium and the sperms. So as a result, the sperms are drawn. As a result, the sperms move towards the sperms are attracted towards the oogonium, and several sperms get surrounded the oogonium. They attach to the oogonium wall. Only one sperm is successful, or one sperm gets successful and enters into this wall of the penetrates the wall of the oogonium and enters the ovum. So the male and the female nucleus. So the male nucleus and the female nucleus, they will fuse, and when it is fused, so the zygote is formed. So this after fertilization, the male and the female nucleus fuse to form the zygote nucleus. So it is the fusion of the male nucleus, the sperm nucleus, and this oogonial wall. So when the oogonial wall is there, so this zygote which is formed, so the oogonial wall will become gelatinized and releases this zygote outside. So the oogonial wall gets disrupted, and the zygote itself gets released into the water. So that means when their conceptacles are present, so they are released in the outer regions, and this zygote, it rests on a hard surface. When it goes on moving, or it is liberated, so when it comes in contact with any solid object, it will rest on that solid object, and it will divide into two cells. So it divides into an upper cell and a lower cell. Now this upper cell and lower cell. So this is the upper cell, and this is the lower cell. So the upper cell gives rise to the thallus. So that means actually the diploid plant is produced from that, and whereas this lower cell gives rise to the rhizoid like processes which help to this is the diploid plant which is produced from the upper cell. So this upper cell goes on dividing to form the diploid plant, and forms the complete plant consisting of the main axis and the primary laterals and the secondary laterals, which further develop the air bladders and also the receptacles. But the lower cell produces some root-like structures, some rhizoid-like structures, which acts like an holdfast to the substratum and helps to fix the plant to that substratum. So that is how the Sargassum reproduces. So and it produces the diploid plant. So the reproduction is very simple by sexual reproduction, that is oogamous. Of course, there is also vegetative type of reproduction by fragmentation, but that fragmentation is not similar to the other types, other examples. Here what happens in the fragmentation, only the young ones, when the old ones, the older parts of the plant, when they get separated or destroyed, only the young ones remain and they continue to grow into a new plant, that is fragmentation. But in sexual reproduction, there is oogamous formation of the sex organs in the structures called as conceptacles. Conceptacles are structures present in the receptacles. So these each conceptacle produces the antheridia and oogonia. In one conceptacle, there are several antheridia produced. Each antheridium is surrounded by two layers, exocyte and endocyte. And each antheridium produces about 64 male gametes which are biflagellates. So these biflagellate sperms are released after maturity through an opening called as ostiole. The oogonium also is produced in the conceptacle. Initially, the oogonium, which is having a single cell or a single nucleus, it divides by meiosis to produce 8 nuclei. Out of the 8 nuclei, 7 of them degenerate, only one will remain and form the egg nucleus or the female nucleus. So this single nucleus, it forms the oogonium, which is surrounded by 3 wall layers, exocyte, mesocyte and endocyte. So that is why the oogonial wall is thicker than that of the antheridial wall. So the sperms which are released, they move towards the oogonium, and many sperms get surrounded by the oogonium, and one of the active sperm will enter into or will penetrate the oogonial wall and fuse with the female nucleus. The male nucleus and the female nucleus fuse to form the zygote nucleus. So this zygote which is formed in the oogonium, so that zygote is released, and that zygote, when it comes in contact with a solid surface, it will rest on that solid surface and it will start dividing by mitosis to form an upper cell and lower cell. The upper cell gives rise to the diploid plant, the lower cell gives rise to the root-like structures which helps to fixation of the plant. So that is about the life history of Sargassum.